Well, hey everybody, I'm Hillary Michelson, and I'm going to talk about some work that we did investigating the inhibitory networks and their contribution to the control of seizure generation and propagation, particularly in the hippocampus. It's highly related to some of the work that Catherine just discussed. So in some ways, maybe it was really helpful that she went first. In some ways, it might have been helpful if I went first to set the stage for her stuff as well. But in any case, when I came into this work, I came from a background of whole animal studies and in vivo recording, behavioral analyses, and sort of looking at epilepsy from a macro kind of perspective. But I became very intrigued with that concept of excitation and inhibition and how these two circuits would contribute to the development of seizure activity. And so I began to focus primarily on the inhibitory network, primarily because everybody else was doing the excitatory system. And so I thought, everybody considered the interneuron circuitry to be a little bit more homogeneous, and I wanted to investigate that further. And that's what I'll be talking about. But before I go there, I just wanted to say that in more recent years, I've started to look at the development of, or maybe the mechanisms involved with the development and prevention of development of post-traumatic epilepsy in a TBI model. And the reason why I wanted to bring that up is not only because it's really exciting work, but also because that particular line of investigation came about from an experience similar to what we're doing here. About 15 years ago, Bob Wong sat down to interview all of the the active investigators that were doing neuroscience-related research as we were establishing the Furchtgott Center for Neural and Behavioral Sciences. And we interviewed all the different investigators, and one of the people that we spoke with was the late Al Stracher, who was doing work on calpain inhibition and calpain inhibitors that would cross the blood-brain barrier. And his work was not related to epilepsy, but it intrigued me enough to start that conversation with him. What about calpain inhibitors and epilepsy? And then from that conversation came a conversation with Doug Ling about post-traumatic epilepsy and models of inducing trauma that we could then implement at Downstate. And from that came a whole line of collaborative efforts with Doug and with Jeff Goodman and now with Sabina and with Lenko Valsamis and a number of different people, also at different institutions, Gatal Hassan as well. And so I guess what I'm saying this for is that I hope that somebody here is as lucky as I was to have their interest piqued about research that maybe they didn't know was going on and begin a collaboration that turns out to be as fruitful as that's been for me. So with that said, I'm going to focus today on really the excitatory synaptic responses that we found that were mediated by GABA, that Catherine talked a little bit more in detail about, and then a little bit more about the interneuron circuit in particular and the heterogeneity that we found within the interneuron circuit, how its connectivity is not at all homogeneous as we had thought. Okay, so to orient you here, we did in vitro hippocampal slices from guinea pigs, and as Catherine was talking about, we used the 4-aminopyridine, which is a potassium channel blocker, as a tool with which to induce stable seizure-like activity in these hippocampal slices. And I recorded from interneurons within the hilus region of the hippocampus, which lays between the two blades of the dentate gyrus. And I simultaneously recorded from pyramidal cells as well. And focus you first here on the pyramidal cells. Here, we did these recordings in 4-aminopyridine, but we also blocked glutamate, fast glutamate channels with inotropic glutamate receptor blockers. And so we were initially looking at this giant iPSP that Catherine spoke about, a synchronous iPSP that had triphasic components, a hyperpolarizing and a depolarizing GABA A component, and a hyperpolarizing GABA B component. But what was intriguing was the interneurons themselves were bursting, and they were bursting in the absence of glutamate stimulation. So what was causing that burst response in the interneurons. We were very intrigued, and so we examined that a little bit further. In the next panel here, what we did was we hyperpolarized the interneuron, and we triggered the response. And you can see with progressive hyperpolarization that we were able to uncover a depolarizing synaptic event that was not mediated by glutamate, but that was driving these burst responses in the interneurons. And that was happening coincident with the giant IPSPs that you can see in the pyramidal cells. When we recorded from pairs of interneurons, we could see that they were bursting synchronously. So that was that synchronous discharge that was occurring in hilar interneurons was extremely intriguing to us, but we didn't know at that time what was driving that synaptic depolarization. When we put in picrotoxin, the GABA A receptor blocker, we were able to block the response in the interneurons, the burst response in the interneurons, and also the individual unitary IPSPs that were also simultaneously occurring. So the depolarizing event that we saw in interneurons was driven by GABA, but GABA was acting as a depolarizing neurotransmitter amongst these interneurons.
2 minutes.
Oof, really? When we washed out the 4AP, you could see, as Catherine was alluding to, that we saw both unitary excitatory postsynaptic potentials and inhibitory postsynaptic potentials that were occurring in the same interneuron, all of which were blocked by the GABA A receptor blocker picrotoxin. And so that was at the time that we did this work, that was done in conjunction with Bob Wong, that was very controversial because GABA was not thought to be depolarizing at that time, as Catherine was saying. It was the inhibitory neurotransmitter. But what we found was that although GABA was depolarizing amongst interneurons, it caused the synchronization of the firing of the interneuron circuit, which then functionally produced an enhancement of inhibition within the pyramidal cells. So that was the first sort of line of inquiry that we pursued. But I was also interested in looking at the circuit in a little bit more detail. And when I recorded from multiple interneurons within the hilus, I found that there were several different types of interneurons that were differentiated anatomically, neurochemically, and also by their connectivity amongst themselves and projections to pyramidal cells. In this slide you can see, or this box you can see, this one particular type of interneuron which fired during the triphasic synchronized IPSP in pyramidal cells. When we washed in picrotoxin, that interneuron was silenced. So that burst was entirely driven by GABA A depolarization. But this interneuron did not contribute to the generation of the synchronized GABA B response that was seen in the pyramidal cells. Another group of interneurons fired similarly to the first before we added picrotoxin, and then when we added in the picrotoxin, the burst was again blocked, implying that this neuron did not contribute to the generation of the GABA A component in the synchronized event in the pyramidal cells, but it also received a GABA B projection. And so, almost done?
No.
Okay. Okay. So fast forward, there was another group that was burst firing. That burst was not sustained by a depolarizing synaptic event, and we followed that through. We stained multiple cells. We found that these cells, if you injected biocitin into one of the cells, it stained multiple cells, implying that these neurons were connected by gap junctions. So there's a proportion of interneurons which are connected by gap junctions, and that electrotonic coupling amongst those interneurons is actually what drives the synchronous triphasic event in 4AP that we see in the pyramidal cells. Because when we washed in carbonoxolone, we blocked the gap junctions and we blocked the production of the triphasic inhibitory response in the pyramidal cells. So just to wrap it up very quickly, carbonoxolone or gap junction, by blocking gap junctions, you could block a synchronized event production in the pyramidal cells. So the gap junctions are actually required to initiate the synchronous inhibitory oscillations that we saw in 4AP. And that's it. That's it.